Hello, my colleagues everywhere in the world. This is Dr. Eze, and today our topic is so exciting. It is not very exciting. Nobody likes epithelium and histology, but anyway, it's a histology topic and it's about epithelium. Okay, and as you know, epithelium is very important. Nobody likes it, but everybody enjoys its results. Everything that appears out of you is epithelium, by the way. So this lesson, it's complicated, but I try to simplify it to the maximum. And I have another surprise for you, which is as follows. We made you some uh, questions in the description. So there are multiple choice questions. And if you could answer them correctly, the answers are there. The right answers are available for you. But if you could answer them correctly, I can almost guarantee you that you are going to do well, either in the papers or in the clinical exams. So before you pass on also, I'm going to be grateful if you could subscribe so you can support me to keep giving this information to your colleagues all over the world for years and years and years. Now let's start with our topic here, and we are going to speak today about the epithelium, as I have mentioned. So our point today is going to be in this page about the definition here. We are going to speak about the key features or key characteristics of the epithelium, and we're going to speak about the functions. So the definition simply, the epithelium is composed of closely aggregated cells. Okay, if you want to remember this one, closely aggregated cells. Okay, and actually those cells or this epithelium has very little matrix, and this is a very important piece of information for you. So they are closely aggregated and there is very little extracellular matrix. This is the key word here. Okay, so they form continuous sheets, as you see in your skin, your membranes, and so on, and they cover the external surfaces and the internal surfaces or cavities. Well, let's move to the key characteristics of epithelium. What are the key characteristics? Number one, its polarity. And here we can say that the cells of epithelium exhibit something called domains. Okay, so the polars or the poles here are called domains. Let me make this easy for you. So let's suppose that this is an epithelial cell. It looks like a pyramid. I'm trying to draw it for you here. I'm not anyway. So here, this is called the apical domain. Here, this is called the lateral domain, and here this is called the basal domain. Okay, so there are three domains of polarity in every epithelial cell. Okay, so the second thing you want to know that the epithelial cells in general are avascular. Epithelium has no blood vessels. It gets its nutrition by diffusion. Okay, this is a very important piece of information for you. The fourth criterion is the basement membrane. Any epithelium in the world is based on an epithelium. Okay, in general, fine, in general. Okay, and finally, they are cohesive. The cells of the epithelium are cohesive. So what does that mean? They are strongly bound together. Okay, and the ways by which they are bound together, we are going to mention. There are many types of junctions, specialized junctions. We are going to elaborate on them later. What are the functions of the epithelium? The epithelium has many functions, but in general, as the skin and hair and nails are all epithelia, it's for protection, right? Absorption, if you consider any place, intestine and other organs. It's secretion also. The exocrine glands, endocrine glands, or contain epithelium. It's, as mentioned in the first page, it's fundamental. It's everywhere. Excretion also, filtration, and sensory reception. We are going to touch on all these later. Okay. So let's start here with the first type of epithelium. It's called the lining epithelium. And before we move on in this, there is something that I want to make life simple for you. Okay, so this classification, this primary classification, is based, as you see here down on the page, is based on the number of cell layers and the shape of the superficial cells. So the classification of epithelium is based on those two factors. I'm going to line them for you. Okay, so first of all, it is based on the number of cell layers. How many layers of cells do we have there? And in addition to that, it is based on the superficial cells. I'm going to elaborate on this later, but those are the two factors that determine the type of the epithelium. So in this way, you are going to see the way how they are classified well. And now I'm going to tell you what are the classes of epithelium before we start. So in general, the epithelium is classified into three branches. So here, this is epithelium, okay, and we have three types. Okay, the first one is the lining one, which we are speaking about now. The second one is called the glandular. Okay, and the third is special types. Okay, this is in general. So each one has subdivisions. Each one has subdivisions. Okay, and each subdivision has its categories. Okay, so we are going to find many other subcategories everywhere. Okay, so this is the general picture here. So let us move back to our lesson here and let us dive in the lining epithelium, and we will go to the subcategories now. The first subcategory of lining epithelium is the simple epithelium. Okay, and just to make the life easy for you, there are only two types of lining epithelium. This is the first one. It is simple epithelium. Okay, so let's move on now. So the simple epithelium is composed of three types. The first type is the simple squamous. And you have to notice here that the simple epithelium, for example, for simple epithelium subcategory or subdivision, you have to mention the word simple, so we can understand what are you talking about. Here, simple squamous, simple cuboidal, simple columnar, simple. You have to mention it's simple. Okay. What is meant by simple? It means that it is composed of a single layer of cells. Period. Okay. So I think that was more difficult for us in the medical college, but it is not really difficult. So you see here, it's composed of single layer. Okay, composed of single layer. Okay, and this layer rests on a basement membrane. Okay, well, so let's start with the simple squamous. There should be a picture here for simple squamous. I don't know where is it now, but I'm going to tell you about this. So you see the first picture up here. Here, this one, the one here. Okay, this one here. So this is simple squamous. To mention the point here for you and make it easy, let us imagine that for simple squamous there is one layer here of ground, and you are making blocks for your room. Okay, granite or something. So it's flat here, and you are making beautiful floor. So this is squamous. Okay. Just remember the floor of your room if you have difficulty in understanding what is simple squamous. What does it mean? Okay. So simple squamous cells are flattened and they are scale-like, and they have centrally placed nucleus. If you have good vision, good sight, you can see the nuclei here in the center of the cells. Okay, sorry for this. There must be a one for squamous, simple squamous, but I apologize for this. It was put, but there is some mistake that happened. Well, what is the function of the simple squamous? It acts in exchange, lubrication, and in barriers, and it's located mainly in the endothelium the of the blood vessels. Okay, so if they ask you what is the epithelial lining of the blood vessels, or what is the tissue type of endothelium, it is simple squamous. Okay, and actually because it is thin, it is and it is based on the basement membrane, it is renewed automatically, so it gets out and it exfoliates and changes constantly. Okay, another type of squamous is the mesothelium lining. So here we are speaking about your internal cavities. Well, so let's go to the simple cuboidal, and from the name here, this is for simple cuboidal, this picture here, as you see here. Okay, and it's cuboidal, right? It's cube shaped, spherical. Okay, here this cube, and as you see, there is a central nucleus, this big nucleus here. This is the nucleus of the cuboidal cell. Fine. Well, what is the function here? The cuboidal cells work in secretions, absorption, and protection. Examples: the small ducts of exocrine glands. We are going to elaborate on exocrine and endocrine glands. What is the difference? Okay, but in simple terms, the exocrine glands has ducts, the endocrine glands has no ducts. That's it. Anyway, the surface of the ovary, you are going to find cuboidal, simple cuboidal tissue, and you are going to find it also in the kidney tubules. There are many tissues and types of the epithelium in the renal system, actually. Well, let's go to the third type or subtype of simple epithelium, columnar, and from the name, the cells are tall. Here you see in this, let me change the color, here this type here, you see. So they are columnar, this tall one, and they are tall, column shaped, and the nuclei are, you see here in the cuboidal, they are oval, but here, sorry, they are circular, but here they are oval. In columnar, they are oval, okay, and longitudinal, by the way, and they are usually near the base. So this is the base down here, and they are on the base. The nuclei are also close to the base. So where do you find the what is the function of the simple columnar? They work in absorption, and usually they are in the microvilli, because you see the villi form the brush borders, and you need let's say white space area, white area for absorption. So the simple columnar is very good for the microvilli or for the intestine, and also we are going to find it in secretions and in protection. Where do you find the lining of the stomach is one of the examples where you can find the simple columnar, and the small intestine, as we have mentioned, in the microvilli, and it's found also in the gallbladder. The second type, as I have mentioned to you, of lining epithelium is the stratified. Now we have simple, and we call everything simple as simple. So simple squamous, simple cuboidal, simple columnar. This is stratified now, and we are going to call everything stratified, and they are the same names, by the way. So they are squamous, cuboidal, columnar again, but they are stratified. What is the difference between them? They are composed of two or more layers of cells. The question is, how can we name them? There we have one basement membrane and one layer of cells, right? They are like this almost. So we know this type. But here, as you see in these images here, we have all this scale of cells, all this scale of layers, okay? Also here you can see all this. So there are two or more layers of cells. How can you name these? And the answer here is you are going to name them according to the cells of the superficial layer. So the up one, let's say the P floor here, Okay, the pantry floor here. Okay, the pantry floor up here. You are going to name it according to it. Okay, so whatever happening inside here, you are going to consider the upper floor. Well, so let us elaborate on this a little bit. I think that I missed the place with drawings, but I'm going to delete everything so everything can be clear for you. Well, stratified squamous. It is flattened again. Okay, and the basal layer is usually cuboidal or columnar, but it is called squamous. Why? Because the most superficial layer is squamous, but the basal layer could be anything, cuboidal, columnar, no worries about that. What is the function? There is something mysterious about this. There are two types of stratified squamous epithelium. One of them is called keratinized, and the other one is called non-keratinized. Where is none? Here it is, non-keratinized. Okay, so they could be keratinized or non-keratinized. So the keratinized stratified squamous epithelium provides maximum protection. That is why it is on the skin or and the dermis too. Okay, the non-keratinized, it's also for protection, and it's for moist surfaces, internal surfaces like esophagus and vagina. Okay, well, where do you find this? As we have mentioned, the skin, oral cavity, pharynx, vagina, and so on. So this stratified squamous epithelium. Let's move to the other type here, stratified cuboidal, and here the image can show you something. Okay, this is the image here. Okay, and in this case here, the surface cell, as we have mentioned, it depends on the, remember this, the surface layer, the cells on the surface layer. So what is the shape of the cells? The surface is cuboidal, and in the columnar, the surface is columnar. Okay, but both of these types are rare. Okay, they are rare. So stratified cuboidal or stratified columnar, they are not common. The commonest type is only the stratified squamous. So this is the one that's going to meet you a lot because the skin is involved here, and the skin is the biggest organ in your body, right? And the body of your patients. Both of these two types actually are used for protection and conduit. And when we tell conduit, we tell ducts, right? We speak about ducts. So here in the cuboidal type, we find it in ducts of sweat glands, for example. Sweat gland is an exocrine gland, right? There are ducts there. So if there are ducts, it's exocrine gland. If there are no ducts, it actually releases its content in the bloodstream directly. It's called endocrine gland, right? So it's usually found in the large ducts of the exocrine glands. Well, we are doing good now. Let's move to the stratified columnar. And in this case here, we find it lining the stomach, and for protection in this case, small intestine and gallbladder. So well, we did well till now, and in this way we have finished the lining epithelium, the first main category. Well, now we are going to the glandular epithelium. And in the glandular epithelium, here, this is the image you see here. Let me just make you something. Here, this is the image, okay, of glandular epithelium. It looks like a gland, something inside, and there are many secretions within, okay? So what you want to know is the definition here. And the definition of glandular epithelium, which is the major second type, cells is specialized for secretion, period, okay? Glandular epithelium is formed of cells specialized for secretion. And in this case here, as we see, they invaginate. This is a part of their development, and this is the key word here. They invaginate from the lining epithelium to form glands. Invaginate. Well, let's move to the classification. And the classification depends on the destination of the secretion. And this is what I promised you about the types of glands, right? So we told exocrine and endocrine, but there are other two types you want to know. The first one is the exocrine gland or exocrine glands. And as I told you, they secrete their products onto the surface for or into lumen by a duct. Anyway, the key word here is duct. See, duct. There is a duct in the exocrine glands. Sweat, salivary, liver bile, whatsoever. Okay. The endocrine glands, as I have mentioned, have no ducts. They are used for hormones. Okay. They directly pass into the bloodstream. Okay. And they are ductless, pituitary, adrenal, and so on. There are other two types of glandular epithelium, and those include the following types. One of them works on the neighboring cells, and the other one works in itself. So they are very simple. The paracrine secretions or paracrine glands, they affect nearby cells, and the autocrine secretions affect the cells that secreted it. So it affects itself, by the way. Okay, independent one. Anyway, let us move now to the third type of epithelium, which is this category contains the special types, and actually these types do not fit down in the page. You will find something written by white color, and these types do not fit neatly into the simple stratified classification but are functionally important. So in some references you may find these two categories included in the main two that we have mentioned, the lining and the glandular. You may find that, but it's okay. It's okay. So now here, just for clarification, they are separated and they are called special types of epithelium. So the first type is called pseudostratified columnar epithelium or pseudostratified columnar epithelium if you are very, very unique person. So anyway, pseudostratified columnar epithelium. From what we have mentioned, we can know that it could be stratified, and if it is stratified, there must be, as we have mentioned, many layers of cells. But they mentioned it is pseudo, so there is a lie here. There is some cheating of some sort. So most probably from the name, it looks like stratified, but it is not going to be. Let us analyze this. And regarding the columnar epithelium part, I think that this one is clear. So let us see here what are the key features of pseudo-stratified columnar epithelium. As I have mentioned, it appears stratified because nuclei are at different levels. So because nuclei are, you see here in the right side here, nuclei are at different levels, some here, but this is pseudo, this is not true. It looks like that only, okay? So you see it, you think there are multiple. When we say stratified, it means multiple layers, okay? So as we are looking here, we think that these are multiple layers, but they are not. Every cell here is one. It is one layer, by the way. So it's simple. It is not stratified. That is why they say pseudo-stratified. It is simple, okay? So they are mentioning here, but it is actually simple. So remember that simple stratified. If they ask you, if the pseudo-stratified columnar epithelium is stratified or simple, you are going to tell them, no. If I have to choose, it is simple, okay? So why? Because all the cells rest on the basement membrane. There are no layers, okay? All these cells that you see are based on a basement membrane here, all of them, okay? Whatever the shape is, they have an end in the basement membrane, okay? Is that clear for you? Well, that's good. Let us go to the last type of our special and of our epithelium in general, which is called urothelium. Okay, and from the name, it is related to the urine or the renal system, and it's also called the transitional epithelium. And actually, it is, by the way, it is stratified epithelium. This is truly stratified, okay? There are layers here, and this drawing or this image here is going to show you. These are actually are true histological images here available for you. So we have many layers. This is apparent here. So as there are many layers, and the basement membrane is down here, so this is considered stratified, okay? Well, so it lines the urinary tract. That is why it is called urothelium. Okay, and the surface cells are called umbrella cells. Surface cells. I'm going to point here. Those are called umbrella cells. Well, and they are large and they change shape. Remember your bladder when it's empty. When it's full. Usually it is stretched, right? These cells are stretched, and when it is empty, these cells are relaxed. So they are large cells and they can change shape. So they have a dome like an umbrella, dome shape when they are relaxed. I think that this is taken for something relaxed here, okay? And when they are flattened, uh, they are stretched most probably, okay? And the reason for that is for the accommodation of the change in volume. So let us read down here. These types do not fit neatly into the simple stratified classification, but are functionally important, as I mentioned before. You won't remember this point. So if you got stuck with classification, or you found someone who tells you is pseudo-stratified columnar epithelium is stratified or simple, you are going to tell. If you are stuck, it's simple. And regarding the urothelium, as I have mentioned for you here, it is considered stratified. Okay, this is the point here. Well, we are going to move now. We finished the epithelium, by the way. If you want to leave now, it's okay. But if you want to get the full information about this lecture or this topic, I advise you to continue. We are so close to the end. Okay, yeah, it is a little bit boring, but we want to know about it. So on the left here, we are going to speak about something that I have told you about before, and I have drawn something like a pyramid, you remember here. And we told that this is, we are speaking about cell polarity. We said this is apical, this is lateral, right? And this is basal domain, okay, or pole or something like this. So in this image here, as you see, this is mentioned in detail in a true cell, okay, through epithelial cells. So you see there is apical domain here, okay, sorry. There is apical domain, there is lateral domain, it is on the sides, and there is basal domain, this histological one. Well, so polarity refers to the morphological and functional differences between the three distinct surfaces, as I have mentioned, or they are called domains of an epithelial cell. So we mentioned an apical domain, it's the free surface. Let's just make a pen here. This is the free surface here, okay. And it faces the lumen or external environment. The lateral domain faces adjacent cells, so they get attached here, as we have mentioned. And as you see, the basal one actually faces the basement membrane, this part here, okay. And actually, it's related to the underlying connective tissue. Well, here we are going to speak about the apical part. We are going to speak about the apical domain, this part here. Is that okay? Well, so there are something called modifications or specializations for the apical domains. Okay, this is very important actually. And if you want to get a high degree or deep knowledge, you have to know this. On these pictures attached for you, you can find these specializations, but we are going to mention them in the text first, and then after that we are going to come to the diagram. So one of the specializations that you are going to find, I want you to follow the text down in white, the microvilli. Other type is something called stereocilia, sorry for this. And finally, you are going to find the cilia. These are the three types of specialization by which modification happens for the apical domain. These are found in the apex or the apical domain of the epithelial cell. So in microvilli, there are small finger-like projections containing an actin core. And this is a very important question in the MCQs. They ask you, what is the core of the microvilli? It's an actin core. Okay, this is important. It seems unimportant, but it is important. Anyway, their function is to increase the surface area, as you see in the right drawing. Okay, so there is a huge area for absorption in this case. Let's say, okay, and it's found in the small intestine and in the kidney tubules. I'm going to use a pen to make it easy for you. We are here, okay, and there's something called the brush border, like this one you see here. This one here, oh, sorry, change the color. This one here, okay, this is a brush border, right? It's apparent for you, isn't? Yeah. So this is the appearance of dense microvilli. So when we have many microvilli like this, it's called brush border. You will find this in the intestine microscopically. Okay. Well, so let us go to this type here, stereocilia. Stereocilia. It is stereocilia. Okay. So we have long mobile microvilli. Function is absorption again, but it is a different type of absorption. I'm going to tell you, it's in the epididymis, okay, regarding the seminal fluids and these things. So for the seminal fluids and epididymis, there is special type of cilia. It's called stereocilia for the epididymis, especially. Okay. So it is also used in the sensory reception, and it's used in the ear, in the inner ear. You use stereocilia. So you can remember it with the stereo of the sound of the music of the this appliance. Okay, it's stereo because it is found in the ear. Okay, well, let's go to the cilia now. And in the cilia, there is long motile hair-like processes contain something called a 9+2 arrangement of microtubules. Don't focus on this. And these are called axonemes. This is something that the histology people want to know. Okay, the function of their function is to move substances along the epithelial surfaces, and the example for these are the trachea and the fallopian tube. All of you know that the ovum rises from the ovary and moves in the fallopian tube, right or not? So we have the ovary here, and we have this fimbri here, and we have this tube here. Okay, so when the ovum comes here, we need these cilia so they can move it down to the uterus, right? So this is another what we can call uh, an apical modification or special. So in simple terms, the epithelial cell has modified itself to do the function because the apical domain or apical side faces the environment and does the outside job. Now we are going to the lateral specialization. You remember the pyramid? We have apical, lateral, and basement. So this is the lateral part here. And down here, actually, these are complexes that bind cells together. So the function of the lateral domains is the binding of cells together. They regulate paracellular transport. It is the relationship between the cells themselves, epithelial cells, and they facilitate communication between the cells. They are typically, we are reading down in white, okay, typically found in a complex called the junctional complex, okay, and usually they are near the apical surface of simple epithelia. So you remember simple epithelium, it's one of the types of the lining epithelium. So in these simple epithelia, okay, near the apical surface, it is not apical, it's lateral, but they are near that part. Anyway, there are many types and there are different locations, by the way. Don't focus on this very well. We are going to detail it. So we are going to move to the first type here. Okay, it is called tight junction or zona occludens. You will find this here also in the drawing. Okay, This is an example for it. Just to have the picture. Here we have adherence junction or zonule adherence, and you can find it here again in the drawing. Okay, we have here again this muosome. This is very important that you want to know. Okay, macula adherence. It is here if you want. Okay, this is dense. Okay, and we have a very important thing that is going to meet you all over your life in action potential, in physiology, everywhere. It's called the gap junction. Okay, I'm going to summarize this fast. We do not want to take much more time. So simply, the tight junctions are the most apical. Okay, they are so close to the apex. And the adherence junction or zonule densa here, you see here from the drawing, you can see this. This is the cell. Okay, so the first one, which is the tight junction, or it is close to the apex. This is the apex here. Okay, so they are close to the apex. The others are just below them here, as you see in the drawing. Okay, and we have this the desmosomes or macula adherens. They could be anywhere. Okay, they are randomly placed, but they could be anywhere. And finally, the gap junctions. Actually, they are gaps only, and they could be anywhere on the lateral domain. Okay, there is more information about this. I do not want to actually make this so boring. So if you want to take more information about this, it is about sealing those lateral domains. So if you want to take more information, all the information actually is already mentioned here. You can stop the video and take an idea about what's going on. Okay, well, let's move to the last slide. And in this case here, we are going to speak about the CAMs. The CAMs. What are the CAMs? The CAMs are the cell adhesion molecules. Those are important. Those are very important. Okay, there is a drawing here. You want to study it by yourself. And again, these are transmembrane protein. I'm going to use a marker here. Okay, the transmembrane proteins crucial for cell-to-cell and cell-to-extracellular matrix or ECM interactions, and they are the key players that form the junctions described above. So what is the job of these CAMs? Actually, they form those here. You remember? Here, those here. I'm going to make it in white for you. Those here. So the CAMs form the junctions on the lateral domains. This is the job of the CAMs, to simplify it, okay? Well, so let's move on here. Okay, the major families of CAMs you want to know, something called caterenes. Okay, cat or C-aterenes, we can call them, or caterenes. They are calcium. If you remember, it's C-aterenes, calcium is involved in this. Okay, so it's for the cells that are identical or molecules that are identical anyway. And there is something called antigreen here. We have caterenes and antigreens, also other type. Okay, and these are actually bind cells to the basement membrane, so they are going to be fixed well on the ground. We have selectins. Okay, they are involved mainly on cell-to-cell adhesion. Okay, transient cell-to-cell adhesion. Okay, especially in the circulatory system. You are going to learn more about this in immunology, and there is something called immunoglobulin superfamily. It's IGSF counts, and those primarily involved in immune response and nerve development. So don't forget the MCQ questions, the multiple choice questions in the description. And if you find this beneficial for you, don't forget to subscribe and support me. Thank you so much for your presence here, and see you later.